This lesson is about the structure of DNA and how the structure relates to the function as a genetic material. DNA is a long polymer made out of nucleotide. Each nucleotide is made out of 3 parts. Again, DNA is a chemical. It's a long chain, which is a polymer, made out of small pieces, monomers called nucleotides, and each nucleotide is made up of 3 chemicals. A phosphate can be written this way, or with negative charges on the oxygen atoms, depending on the pH of the solution. The second part of a nucleotide is a sugar. For DNA, that's deoxyribose, and for RNA, it's ribose. So we'll say deoxyribose. Ribose was discovered first. Ribose is a 5-sided sugar, 5 carbons. They're numbered starting at this point: 1, 2, 3, 4, 5. So that's ribose. Deoxyribose was discovered next, and it looks like this. And again, the only noticeable difference between the two is, yes, you got it, deoxyribose is missing the oxygen attached to carbon number 2. Hence the name deoxyribose. The third part of a nucleotide is a nitrogenous base, or a nitrogen-containing base chemical. Each of these chemicals is a carbon skeleton with nitrogens attached to it. Some have 2 rings, others have a single ring. The ones with 2 rings are called purines. The ones with a single ring, pyrimidines. And it turns out that the purines, these chemicals are adenine and guanine, which abbreviated with the first letters A and G, and the pyrimidines, thymine and cytosine, which are abbreviated T and C. To make a nucleotide, a phosphate molecule is connected to a sugar, in this case deoxyribose, using an enzyme that will dehydrate a water molecule away from the phosphate and this hydroxyl group hooked to the sugar, and leave the 2 linked together like this. The next step is to attach the base, the nitrogenous base, to the hydroxyl group at the 1' carbon of the deoxyribose sugar. And I'm not going to draw the molecular shape of adenine or guanine or thymine. I'll just use the abbreviation, the letter abbreviation. In this case, adenine connects. So this is a single nucleotide, a phosphate, a sugar, and a base. To connect these nucleotides together as a polymer, if 2 nucleotides are brought near each other, and then again an enzyme can take a hold of these 2 nucleotides and dehydrate these 2 together at this point, producing water, then the 2 nucleotides stick together, as shown here. Now we have 3 nucleotides hooked together. One of the most significant rules in DNA structure is that the purines pair up with pyrimidines in a specific, very specific setup. Adenine always binds to thymine. Guanine always binds to cytosine. And they do so with hydrogen bonds. The shapes of the molecules just happen to fit with hydrogen bonds. Adenine and thymine form 2 hydrogen bonds between them. Guanine and cytosine 3 hydrogen bonds. Now you can see if we have nucleotides starting at the top here and going this direction, 3 nucleotides, we could add another nucleotide to this point here and just continue on, add a 4th, a 5th, and just keep making this chain longer and longer. It can be millions of nucleotides long. However, DNA has 2 halves. The other half, the bases T, G, and C, are connected to their sugars. However, I can't have the sugars facing up in the same direction, because then the phosphates would hook on the left side instead of the right. So, to solve that problem, if I turn the sugars upside down like this, then it will all work out. And we have our first upside down nucleotide attached to the opposite side. And we do that with each strand. So the opposite half of the DNA runs to the first half So the two strands are parallel but running in opposite directions like two lanes of a divided highway. So we call them anti-parallel. And that's how the molecules of DNA line up, so that adenine always binds to thymine, guanine always binds to cytosine, and because they're anti-parallel, it forces the molecule to twist around itself in that very familiar shape, the double helix. Now, to save time, as long as scientists remember that DNA actually has phosphates, sugars, and bases, and the phosphates and sugars are always hooked together, they can just write phosphate, sugar, base, phosphate, sugar, base, phosphate, sugar, base, phosphate, sugar, base, and just keep going on and on and on. Of course, on the opposite sides, you'd have the complementary bases connected to their sugars and phosphates, forming the familiar ladder-looking shape of DNA. And this is an abbreviated way to write DNA. These sections here, the phosphates and sugars, are called the sugar-phosphate backbones. And if you think about a backbone of a skeleton, what's the purpose of a backbone? Well, it's to hold everything together. I mean, look at what attaches to a backbone. You got the skull, the shoulders, the hips, the ribs, everything connects to the backbone. That's the purpose of these sugar-phosphate backbones in DNA, is to hold the molecules in the right positions where they're supposed to be. The important parts, the parts that code for the information, are the bases. So the center part, these are called the bases or base pairs. And that's where the information is. Since the important part of DNA is the bases, oftentimes you'll see it just written as the bases, ignoring the sugars and phosphates completely. Now, always remember that the sugars and phosphates are there, but the information is encoded in the bases. And if you know one half of a DNA strand, because of the base pairing rule, the complementary base pairing rule, you can automatically know the other half. For example, on this one, so you should be able to predict, if given one half of a DNA strand, what the complementary half will automatically be. The information in DNA is actually in the order of the bases, similar to information in the English language. Our language is composed of 26 letters in an alphabet, but the order of the letters makes all the difference in the world. For example, if you read the following letters in this order, it brings to mind a certain image. However, if you change the order of the letters, let's just change 2 letters, then suddenly you're thinking of giant New York City rats or something massive like that, maybe rodents of unusual size. See, the order of the letters makes all the difference in the world. The same is true for DNA. If, for example, the DNA letters say ACCAA, that might be the instructions to start building one protein, a certain protein, maybe hemoglobin. If, on the other hand, we change these letters, AAACC could code for a totally different protein. Could be completely different instructions, and that could cause major problems. I mean, you wouldn't want, say, hair growing out of your liver. So that's how the structure of DNA relates to its function as a genetic material.